Very warm welcome to our biology class. So in this class we are going to understand regarding the meiosis, because before understanding the meiosis you are not properly able to understand how the spermatogenesis occurs and how the oogenesis occurs. That things are not able to properly understood if you don't know regarding the meiosis. Therefore, we'll first understand the meiosis in this class today. So you might be observing here the boy. Once suppose this boy has reached to the age of maturity, means by the 12 year or 13 year, or in some individuals it may be by the 14 year, what will happen in his testes, inside the seminiferous tubules, whatever the germinal epithelial cells are there, they start to divide. Even you are observing here the female. In female, inside the ovaries, whatever the germinal epithelial cells are there, they will also start to divide. But the difference between the male and female is germinal epithelial cells will start to divide in male whenever they reach to the age of maturity. But in case of female, germinal epithelial cells will start to divide immediately once the ovary has got originated inside the fetus body. Means by the completion of the third month of the gestation itself, all the germinal epithelial cells will start to divide and they add number of oogonia. Now listen here students, whatever the germinal epithelial cells are there, either they may be in the male testis or they may be inside the female ovary, but these germinal epithelial cells will start to divide and add number of oogonia, and in those oogonia meiosis occurs. And remember that only these are the cells in either male body or inside the female body, they are dividing by the meiosis. No any other cells in the both male and female will divide by the meiosis. Rest of the every cells, suppose for example here the Sertoli cells are there, and inside the, or even in the entire body also, there are the many more cells are there, but all those cells will divide by mitosis. But these cells are only going to divide meiotically. But during the initial stage, they even divide by the mitosis. We will see that when they are going to divide by the mitosis by considering one of the germinal epithelial cell. Listen now students, either you may consider the one germinal epithelial cells from the testis or from the seminiferous tubule. You may consider the germinal epithelial cells either from the ovary also. So by taking one single germinal epithelial cell, we will see that what exactly going to happen now. Okay, suppose now I have taken here the one germinal epithelial cell. We'll see even what are all the conditions inside it. That germinal epithelial cell we have taken from the either from the seminiferous tubules of the testis or from the ovary, you may consider here. Now this is the germinal epithelial cell and it is a normal cell students. If it is a normal cell, then whether every cells are have a plasma membrane or not? Yes, even it also have a plasma membrane, and inside to the plasma membrane we'll find the presence of nucleus here, and there is a nuclear membrane also there, and in between the nuclear membrane and the plasma membrane we will find the presence of cytoplasm also. But inside the nucleus we used to find the presence of chromatin plus nucleolus. In a PUC first year cell cycle cell division, they have clearly mentioned the nucleus contains the nucleoplasm in which it contains the chromatin and nucleolus. Now mainly we'll focus on the chromatin. What are the chromatins are there? To what we must call the chromatin also. You might be observing one of the thin thread-like structure, elaborated structure here. So these, whatever the thin thread-like structures you are observing, these are nothing but the chromatin only. Now we'll magnify one of the chromatin to understand how structurally it is made up of. It is what the one thin chromatin, and this chromatin have one primary constriction here. Yeah, there is a primary construction. So this, whatever the primary construction is there, it is called as an centromere. And centromere have the centrally placed one of the small dot-like structure, and that is called as a kinetochore. And this, whatever the entire thin elaborated thread-like structure is there, it is called as an chromatid. Got it? So this is the chromatin. Don't at all get confused about the chromatin and chromatid, all these words, students. Chromatin means what? So these are the thin elaborated whatever chromosomes are there, we call them as an chromatin. Suppose, for example, one of the cell, whenever it is not dividing, suppose by that time the chromosomes are present in the form of chromatin only. In your body, whatever the cells are there, all those cells are have a chromatin. If they are not dividing, suppose whenever they start to divide, by that time only condensation of the chromatin occurs, and then we need to call them as an chromosomes. Till that, we should not call them as an chromosomes. But usually what happens, most of the time we used to say that every cells are containing the 46 chromosomes, we say. Actually, they are not present in the form of chromosomes. They are present in the form of chromatin. Have you understood how the chromatin is? One single chromatin, if you taken, it is a thin thread-like structure, and in one place there is a presence of primary construction. That primary construction is called as an centromere, and this is the chromatid. Don't consider again the chromatid here too. Yeah, means you should not consider like this is the one chromatid and this is the one more chromatid. No, this is the entire one chromatid only, and this is the entire one chromatin only. So then, how many such chromatins will find inside the cell? Altogether 46 such chromatins are there. It means we usually say that 46 chromosomes are there. Means chromosomes are present in the form of chromatin now here in this particular cell stage. It is the germinal epithelial cells. I hope you are remembered what I told shown in the previous slide. So these are the germinal epithelial cells. You may consider from the ovary also, you may consider it from the testis, but these are the germinal epithelial cells now. Okay, I told total 46 chromosomes are there. Then how we used to represent it? We used to represent it as a 2n. 2n means the diploid condition. So diploid chromosomes are there. Diploid chromosomes in case of the human being, it is equal to 46 chromosomes. It may not be 46 in all other organisms also, but in human, total 46 chromosomes are there. Suppose in his body, whatever the number of cells are there, in every cell except matured RBC, all those cells are have a nucleus, and in that nucleus, total 46 chromosomes are there in every cell. In the same way, in female body also, except matured RBC, all other whatever the cells are there, the cells may be in her leg region or in maybe in the body region, in maybe in the face region, inside the eye, wherever they may be, but in every cells are have the total 46 chromosomes. Okay, my question right now, this particular boy have a 46 chromosomes in his every cell. From where he has received? Or take for example this girl. This girl has even total 46 chromosomes in every cell. From where she has received? Of course from the father and mother. Right? You can observe here. 46, and usually we call it as a 23 pairs. Yes or not? What does it mean by the pair, students? Most of the time we say pair. Yeah, one pair, two pair. Pair, whenever we say pair means they are present in a two copies. Yeah. Now, suppose for example, this will consider this red colored whatever the chromosome is there. I have taken here chromatin. I have drawn here. Now, so this is what the one more chromatin. We can say both together are the first pair. Like this, how many pairs are there? 23 pairs are there. Suppose this is the second pair I have drawn here. This is the one chromatin and the another chromatin, both together forms the second pair. Like this, if you keep on observing, total 23 pairs of chromosomes are there in our every body cells. I want to tell you something, students, here. Whatever the 23 pairs are there, out of that 23 pairs, 23 copies means this set, suppose this set you have received from your father, and another set of 23 are received from your mother. This is what the way how you even received in your body cells. Suppose here this boy have 23 pairs of chromosomes in his every cells except matured RBC. It means that out of that 23 pairs, out of that 23 pairs, 23 are received from the father, 23 are received from the mother. Here also 20 in her also 23 are received from her father and 23 are received from her mother. That is what the way, students. Now suppose for example this is the first pair I said. Now if you consider both these first pair, these are even called as an homologous chromosomes. Yeah, one pair, it is the homologous chromosomes. Yeah, and this is the one more homologous chromosomal pair it is. And in the same way we can use to find the presence of 23 homologous chromosomal pair. Got it, students? I will tell you some even difference also in case of the male and female. Whatever the 23rd chromosomal pair is there, it is not called as an homologous chromosomes in case of male, because XY condition is there. It is the pair of allosome. That's why it is the means not a homologous. Yeah, it is hemizygous condition is going to be appear in case of the male. Now observe here, I said this is the first number of chromosome received from the father, and this is the first number of chromosome received from the mother. Both together they forms one homologous chromosomes, or first pair they have formed out of 23 pairs. Listen very carefully, and it is very basic also, you required it too much. Now whatever these both the homologous chromosomes are there, remember that both those homologous chromosomes are contains the same type of genetic makeup, and they are controlling the similar type of characters in the either male or in female body. What does I mean? Suppose for example the nose shape maintaining gene is present located on the chromosome number 1, suppose, which is received from the father. The same gene even located on the chromosome number 1 which is received from the mother. Means whatever the character which is controlling by the gene chromosome which is received from the father, same character even controlled by the chromosome which is received from the father. Then you might be confused now, sir, why we need to have two copies of every chromosomes then? If this red color chromosome is doing some work and the exactly similar works are going to be controlled by this another white color chromosome also, then what is the use of having two copies here? Yes, chromosomes are present in two copies, students. That's why we even say that yeah, alleles are there. Yeah, alleles means what? Alternative form of genes. So they are present on, one is present on this and another one will be present on this. You will understand about these alleles and how these are homozygous, heterozygous conditions all in a very better way in the next one more chapter. In principles of inheritance you will understand, as well as molecular basis of inheritance, where also you are going to understand regarding what these alleles means, what these genes means, all that detail you are going to understand, students. I hope you are got clear about it. It means that in our every body cells we have a total 23 pairs of chromatin, shall I call it a chromatin, and out of that total 23 pairs of chromatin, means 23 are received from the father, 23 are received from the mother. But in case of male, whatever the 23rd pair is there, it will be XY. And in case of the female, it will be XX chromosomes will be there. In case of females, we can say, in case of male, total 22 homozygous chromosomes are there. And whatever the 23rd is there, it is called as an hemizygous condition. So what does it mean by the hemizygous condition? We'll discuss in one of the other class, but right now we are unable to discuss about what homozygous, hemizygous, all that things, students. Yeah, I want to make you to focus on particularly meiosis. Yes, slowly I'm taking you towards the meiosis. Before that, I'm giving you the perfect basis regarding all the germinal epithelial cells, what happening inside them. Right now, here I told you nuclear membrane is there, and in between the nuclear membrane and plasma membrane we used to find the presence of cytoplasm is there, like it is a normal cell, students. And every cell even have one more structure here, it is called as centrosome. Do you know that how the centrosomes are made up of? They are made up of the centriole. Suppose this is the one centriole and one more centriole will be remains perpendicular to each other. So, and that will forms the centrosome. Means the two centrioles are comes together and they remains perpendicular to each other. They forms the centrosome. This is what the one centrosome is also present in every animal cells. Plant cells don't have this type of centrosome. There is a different type of the condition is there. And in addition to that, students, in this germinal epithelial cell also we used to find the presence of mitochondria, Golgi complexes, yeah, and even endoplasmic reticulum, right? So all these are the normal cells only, like it is not, it is like the normal cell only. I hope you understood now. This is what the way how the germinal epithelial cell is present inside either seminiferous tubule in case of the male you may consider, or inside the ovary that may you consider in case of the female. Got it clear? Now what happens with this germinal epithelial cell? It need to be divided first. Yeah, how it is start to divide? Actually, it will first divide by the mitosis, not by the meiosis. Please remember here, germinal epithelial cells are only the cells they will divide by meiosis, of course, but before going for the meiotic division, their number is going to increase. Germinal epithelial cell number is going to be increased. How it is going to increase? It is increased by the equational division. It means that it is mitotic division is going to occur in that will lead into the increasing the number of germinal epithelial cells. But whatever the divided and newly formed cells are there, we call them as an spermatogonia in case of male and oogonia in case of female. You can observe here they will divide by the mitosis, and this cell has a divide and forms the two more cells. Again, these cells will also once again they will divide by the mitosis only, and they leads into the formation of four more cells. I am talking about simply only one cell, and whatever all newly formed cells are there, they are the spermatogonia in case of a male inside the seminiferous tubule, or you may call them as an oogonia in case of the female. You probably understood it. And one more very important thing is, whatever the spermatogonia formation and addition is there, in case of the male, it is going to occur only whenever they reach to the age of maturity. But in case of the female, whatever the oogonia formation and addition is there, it is occurs immediately after the formation of the ovary. That is the major difference between the male and female characteristics, yeah, in between the spermatogenesis and oogenesis, that what we can say. Right now we want to consider one this particular spermatogonia, or you may consider it one as a oogonia, and then we want to know that how this oogonia is going to divide. Have you remembered, students, this oogonia, once the oogonia form, that oogonia will become enlarged and we call it as an primary oocyte in case of the female, and in case of the male, spermatogonia will go through the growth and it is going to attend at the larger in size, and we call it as a primary spermatocyte. Yeah, in case of the female we call it as a, after the growth we call it as a primary oocyte. In case of the male we call it as primary spermatocyte. We'll see that how it is going to take place now. Yeah, this whatever the entire phase is there, students, we consider it as a multiplication phase. Have you remembered that in the spermatogenesis and in the oogenesis, first phase is the multiplication phase? Yes, this is the first multiplication phase. In case of the male, this multiplication phase is going to occur whenever they reach to the age of maturity. But in case of the female, this multiplication phase is going to occur immediately after the formation of ovaries inside the female fetus only. Right? Now, we will understand the next one here. We'll take one of the spermatogonia now. Yeah, we'll consider one of the spermatogonia, or you may consider it as a one of the oogonia. Yeah, means it is a continuation from the earlier slide. Yeah, here I have shown all this simple cell. Once again, the same cell oogonia I have drawn here. You can observe now. It is what the oogonia and or spermatogonia. It is diploid in condition. It means that it have total 23 pairs of chromosomes. Total 46 chromosomes are there, and one of the centrosome is also there. Now, this whatever the cell is there, it has to go through the G1 phase to initiate the meiosis. You can observe here, students. Yeah, to initiate go through the meiosis, first even it need to go through the G1, S, and G2 phase. Yeah, now first we will understand how and what changes are going to occur in a G1 phase with this spermatogonia or oogonia, what we have considered here. Now, what happens, students? During the G1 phase, the cell is become highly metabolically active, and whenever it is become highly metabolically active, by that time it is going to produce large amount of cytoplasm. Yeah, you can observe everything is similar, but the one major thing is happened here, the increase in the cytoplasmic volume. Yeah, the volume of cytoplasm has increased. After this complete increase in the cytoplasmic volume, what we call it, primary spermatocyte in male, primary oocyte in female. Are you got it? This is what the way how the primary spermatocyte and primary oocytes are going to be produced. Yeah, fine. Now we will take again consideration of this one single chromatin, and we will understand about it. Yeah, no any changes, students. Everything is similar here. Yeah, because just it has gone through the growth phase. Nothing has changed along with these things. Because of that, I have just taken it as it is and copied here. Yeah, but only what changes are occurred from the spermatogonia or oogonia to become a primary spermatocyte or primary oocyte, the change in the volume of cytoplasm. Yeah, that change in the volume of cytoplasm leads into the leads into the increase in the size. Yeah, now you can observe, students, this what we call it as a growth phase. In spermatogenesis also, in oogenesis also, second phase is growth phase. First phase is the multiplication phase. What happens during the multiplication phase? I have shown already. Yeah, this is the growth phase. In a growth phase, what happened? The size has got increased. Now, listen very carefully. The increased in the volume of cytoplasm, yeah, changes in the cytoplasm and nuclear volume ratio. Yeah, suppose if you taken the here, okay, you just observe this cell right now. Yeah, this whatever the nucleus is there, it is the one which is controlling the activity of all the entire cell. Yeah, it means it is properly controlling the activity of all entire cell here. Right now, what happened? Cytoplasm has got increased because the cell has become metabolically active, and there is no changes in the nucleus. Because of that, what happened? The nucleus need to be go through the division now. Otherwise, what will happen? This nucleus will not able to control the function of entire increased cytoplasmic content. Means the whatever the cell organelles number has increased or cytoplasmic volume has increased, so that all the things are not able to remains controlled under the nucleus, because the nucleus has started to remains. Ratio is going to change, students. Means the nucleoplasm ratio and the cytoplasmic ratio, in between the nucleoplasm and cytoplasmic ratio, whatever the normal ratio is required, that ratio is going to be altered. Because of that, the cell now must have to go through the division. Either that division may be the mitotic division or it may be the meiotic division. But here this is the germinal epithelial cells, and it forms the oogonia. Means the spermatogonia or oogonia is there. It must have to go now through the meiotic division only. There is a no any option for this cell. Yeah, it leads into the cell division. Got it, students? But yes, here cell division is going to occurring here. Now what phase of the cell cycle has completed? G1 has completed. Where the cell must have to enter now? S phase. Oh, do you know that what happens during the S phase? During the S phase, DNA replication is going to takes place. We'll see here. Yeah, in this particular slide, we will observe that what things are going to happen. You can observe here. It is the primary spermatocyte or primary oocyte. It have a means a total 23 pairs of chromatins are there. Yeah, and all. This is what the structural details, students. This is the G1 phase. I told you cell is metabolically more active by this time, and protein synthesis is going to increased by several fold here, and there is a even increased in the ATP synthesis also occurs. After this, the cell is going to started to continue in the next phase. Next phase is the S phase, that what I told you. From the G1, it must have to enter into the S phase. You can observe here what happens during the S phase. Yeah, you first have to focus on the chromatin materials first. Yeah, so because of that, what I will do is, okay, I'm going to draw all those earlier chromatin like this. Yeah, because make you to convince properly regarding all these structural changes in the chromatin, that I have drawn it like this. Same thing, but I have drawn it like this because I want to show you the changes occurring which in between the chromatin. Now, are you able to observe here what changes are occurred? Every chromatid has formed its one more copy of whatever the changes are. Structure is there, students. Whatever the structure is there, everything is copied once again here. You might be observing two copy. Red has become one more copy. This blue has become one more copy. White has become one more copy. This chromosome also become one more copy. Shall we once again focus by enlarging the one of the chromatin here? You can observe, students, during the S phase, yeah, histone proteins are synthesizing, DNA replication is occurring, and even the centrosome get replicate also here. You can observe, yeah, centrosomes are also even got replicated. Here there is no replication. If anyone asks you the question, during what stage centrosomes are going to get replicate? So during the S phase only, centrosomes are also going to get replicated. See here, usually during the mitosis, only one time replication is going to occur. Means one is going to form the one more copy. But during the meiosis, it is going to divide once again. Means the total 4 copies are going to be resulted by that type of the replications during meiosis. But during mitosis, only one time replication is going to take place. That is the difference is there during the replication of this centrosomes. Now we will understand the structural details of this chromatin also now. See here, for example, this is the earlier chromatin and it has formed the one more copy also. Yeah, you can observe here. It was the original chromatid. Now it has formed the one more chromatid, but to that one more newly formed chromatid, we call it as a sister chromatid. And exactly at the center, again the centrosome is there here, and we used to find the presence of one more structure, that is a kinetochore. Here also I have shown the kinetochore, students, but even the kinetochore has replicated, means the 2 copies of the kinetochore are formed. One in on this side, one is on this side. You must have to remember this, students, because at this place only spindle fibers are going to get attached at the time of cell division. It is the most important structure for the chromatin or chromosomes, which helps in leading the chromosomes on the towards the opposite pole, right? It is the one more kinetochore is also formed here. Yeah, got it. The kinetochore plays a very important role. This is what the way how the chromatin, even it is in the chromatin form. Yeah, even it is in the elaborated form only. It is not yet condensed here right now also. It is a thin thread like. Just I have made it the one magnified here. Yeah, now where the cell has to go now. Yeah. Once again, shall we see here what happened during the S phase? DNA replication has occurred mainly, but to DNA replicate, it needs the histone protein synthesis has increased, as well as centrosomes are got replicated. That what the events which were occurred during the S phase of a cell cycle. Now after this, if you observed the content of the DNA, whether the DNA content has increased or not, students. Yeah, earlier here if you consider doing the Z1 phase, DNA content will be considered as an 2C, but right now whether it has become double than that, it is the 4C. If you want to understand regarding the what 2C and 4C, you have to understand it like this. Earlier you might be observing here, this is what the chromatin, but now one more copy has formed. It is considered as an 2C, it is considered as an 4C. Yeah, when the cells are become a 4C, then during the S phase. And after this, the cell is going to get into the next phase. As you are observing here, it is the G2 phase. Now we'll take during the G2 phase. What is the changes during the G2 phase, students? No absolute any changes. The cell will only prepare the something which is already even happened during the G1 phase. Same things are even going to be repeated here. You can observe, so these are all the centrosomes are there here. Cell is metabolically more active by this time, protein synthesis occurs by this time, and increase in the ATP synthesis is also going to takes place. Now this whatever the primary oocyte is there, now here also we should have to call it as a primary oocyte, or you may consider as a primary spermatocyte. Now this primary spermatocyte is ready to enter into the meiosis. Yeah, got it. Means G1 completed, S completed, G2 completed, and then it is entering into the meiosis. If it is the mitosis, then so by that time the cell after the G2 it is going to enter into the mitosis. But now as it is a spermatocyte, primary spermatocyte, or it may be the primary oocyte, then it must have to enter into the meiosis. What happens during the meiosis? There is a meiosis 1 and meiosis 2 are there, and in a meiosis 1 there is a prophase 1, metaphase 1, anaphase 1, telophase 1, that substages are there. Again same phases are even going to be found during the meiosis 2 also, and but meiosis 1, prophase 1 is the very important phase where the many more events are going to take place, and it is a very important for the sexual reproduction also, where the genetic material is going to exchange. So in my next video, I'm going to explain you the events which were occurring during the meiosis 1, prophase 1 particularly. Yeah. So you, without missing, you have to observe the next video that will really help you to understand what during the meiosis 1, prophase 1. And in the prophase 1, there are the 5 more substages are there. Yeah, like leptoptin, zygotin, pacetin, diploptin, and dikinesis. All those subphases are there. We will understand all those things in very detailed way in the next video. Without missing, you have to observe it. Yeah, I hope this video has become useful for you all. It has become this video is going to become a little bit lengthier. So because of that, you have to focus this up to the end, so you will be able to get the perfect idea about the cell division. Thank you, students. Thank you once again for watching this video, and please keep on watching. And even if you not yet subscribed, please do subscribe my channel, and even share with your friends, as well as like it and write your valuable comments also. Yeah, please give your like, students. That will really motivate me to work hard in the future days. Thank you once again, students. Take care.